This method can help answer key questions in chemical biology and drug discovery, such as does your compound bind to the target protein?
The main advantages with this technique are that there's no requirement of detachment of the adherent cells, and spatial localization can be determined by imaging.
Though we have applied this method in cell lines, it's also possible to apply this method to other cell systems, such as primary cultures and co-cultures.
Before seeding the cells, place black 384-well imaging assay plates in a tissue culture hood and cover the plates with aluminum foil before using a standard drill to make 3.5 mm diameter holes on both ends of each plate. When the plates are ready, use aseptic technique to wash an A431 cell culture with 5 to 10 milliliters of PBS and incubate the cells with 2 milliliters of trypsin at 37 degrees Celsius until the cells detach. After counting, dilute the cells to 5 times 10 to the 4th cells per milliliter in culture medium and seed 40 microliters of cells to each well of each assay plate, gently shaking each plate from side to side after seeding to ensure a homogeneous distribution of the cells across the well bottoms. To minimize plate edge effects, allow the cells to settle at the bottom of the assay plates for 20 minutes at room temperature in the back of the laminar flow hood before placing the plates in a custom humidified chamber for 2 to 3 days at 37 degrees Celsius and 5% carbon dioxide. On the day of the experiment, use a plate washer to aspirate the medium from each well.